Big data, health informatics, and bioinformatics are some of the buzzwords that you have heard these days. What is bioinformatics? Why is it important? And how can we use bioinformatics to solve some of the problems that we face every day? These are some of the questions that we will attempt to address in this lecture. In 1799, while constructing a fort in Egypt, a group of French soldiers came across an ancient Egyptian tablet containing hieroglyphics on it. What made the discovery this tablet so groundbreaking was not just the fact that it contained hieroglyphics, because by this point quite a few stones without script had already been discovered. It was what came with the hieroglyphics that made the stone so important to Greek. Because while for over 1,000 years, no one has been able to read the ancient Egyptian script, but everyone trained in classical languages could read Greek. This discovery 40 years later, enabled the French scholar to find a way to decode the meaning of the hieroglyphics. The symbols which have been silent for over a millennium became a new window into ancient Egyptian life and culture. Today, we are faced with a challenge and an opportunity similar to that of the Rosetta Stone. It comes from a language which works in any human language in the immense number of volumes written in it, and that is the language of DNA. DNA contains the blueprints for every biological process of every living thing on Earth, and even a small defect in the genetic code can cause devastating health consequences. Understanding how genes work, both individually and with each other, is crucial to the development of the 21st century personalized medicine. However, the problem is that, even when we actually have an organism's DNA in front of us, we are not possible to sift through it with an army of people to understand it. It is often not easy to tell when an individual gene does. Even more difficult is to tease out the subtle influences that multiple genes have on each other. But, answering those kinds of questions is foundational to attacking genetically influenced diseases at the root cause. Today, new high-throughput technologies bring unprecedented opportunities for life science research. They allow us to get new data never seen before, to study new questions impossible to study before, and to discover new phenomena unimaginable before. Now we can sequence not only one person's genome, but also many different people's genomes. This allows us to study the genetic differences between different people at genome scale, as well as many other questions in population genetics. At the same time, we can also study the genetic differences between patients and normal controls to find the genetic mutations responsible for some of the diseases. In addition to the human genome, over 1,000 trillion base pairs from over 165,000 species had been sequenced by the end of 2013. Just reading them would take 30 million years. Life science data is not only big in size, but also growing exponentially. This figure shows the number of nucleotides sequenced and stored in the GenBank database over year. The number of nucleotides had been increasing exponentially since 1982, doubling every 20 months. What does this mean? It means that the data deposited into GenBank in the next 20 months from now on is as much as all the data that have existed in the history of human till today. At the same time, the sequencing cost had been decreasing every year. The past few years had seen even steeper growth of sequencing data. The reason is that next-generation sequencing technologies had been developed and used more and more widely in all areas of life sciences. Next-generation sequencing technologies can sequence one person's genome in a day with less than $2,000. Just think about what exciting discoveries these never-seen-before data may contain. Other than sequencing data, other high-throughput technologies such as proteomic, metabolomic and etc. had also generated a large variety of data. With great opportunities come great challenges. The huge amount of data requires efficient methods. Exponential growth requires scalable methods. The low signal-to-noise ratio requires accurate methods. And handling multiple types of data requires data integrative methods. These are significant technical challenges. But, it depends on how you look at it. Technical challenges can also mean opportunities for technical innovations. The birth and growth of the field of bioinformatics has been driven by these opportunities and challenges. As you may be able to realize by now, the integration between life science and computer science is inevitable, and the result is the birth and growth of the field of bioinformatics. Bioinformatics can be defined as an interdisciplinary field that develops and applies